Welcome to the Deep Dive, where we take your source material, strip away the jargon, and get to the insights that make you truly well-informed. Our mission today is to explore a field that is, well, it's completely redefining medicine. Cell and tissue engineering. We're talking about its applications in medical biotechnology. We're going to be talking about growing organs and repairing what was once thought to be irreparable. And to help us navigate this really intricate landscape where biology meets engineering, we are so honored to be joined by technologist Dr. Rama Yusvana. He's a senior lecturer from the Faculty of Engineering Technology at Tun Hussein on University of Malaysia, UTHM. Thank you so much for guiding us today.
It's a real pleasure to be here. If you think about the bigger picture, the excitement really comes from realizing that these engineering principles, things like precision, material science, structural design, we can now apply them directly to biological failure. We're moving away from just managing a disease.
Their actual restoration.
It's restoration, exactly.
That's a perfect way to put it. And for you listening, that's our journey today. We're going to start literally at the surface with bioengineered skin. Then we'll dive deeper into structural repair, focusing on bone regeneration. And finally, we'll tackle that monumental challenge of creating artificial functional organs. We'll even look at the bioartificial pancreas. So, okay, let's unpack this. Let's start with the skin. When you think about this organ, early attempts to replace large sections must have struggled horribly with, immune rejection and integration. Where did the crucial scientific innovation really begin?
That's a great question, because you're right. The early challenges were immense. The body it instantly rejects foreign tissue. Historically, the innovation was driven by necessity, specifically the huge demand for treating severe burns during World War II. While some early 20th century studies looked at the immunologic basis of grafts, the real breakthrough technology, it emerged in the 1940s and 50s.
Okay, so that's the time frame.
Right. Scientists like Peter Medawar, Billingham, and Reynolds, they started experimenting with what's called in vitro epidermal cell culture, basically showing we could grow human cells outside the body. And what's fascinating is how the technique evolved. From the 60s to the 70s, the focus shifted to adding growth factors.
The biological instructions.
Exactly. The instructions to make these cells grow quickly and reliably enough to actually be used in a clinic. And that all culminated in 1979 when Green and his team successfully transplanted large epidermal cell sheets onto patients.
Wow.
That single act, really, it paved the way for commercial products to start appearing by 1990.
That is such a rapid transition from lab to market in just over a decade. It really highlights the critical need. So if we look at a commercial substitute, something like Apligraf, what is an engineer actually building? How does that compare to our own skin?
Well, we're essentially building a simplified but highly functional scaffolding system, something the body can recognize and integrate with. In a product like Apligraf, the process starts by creating what's called the dermal matrix. That's the structural base.
Okay.
And we make that using human dermal fibroblasts and specific connective tissue proteins. Then that matrix is seeded with human epidermal keratinocytes, which form the outer barrier layer.
So it's a two-layer structure.
A two-layer structure,
Okay.
keratinocytes, fibroblasts, and the dermal matrix. The key insight is that while our own skin is incredibly complex, it has melanocytes, lymphocytes, all sorts of things.
Right.
The bioengineered version targets only the essential structural and barrier components. It provides a biological dressing that's also a template for real healing.
A template for healing, not just a static cover. That's the key. And this is where it gets really interesting, right? The real-world impact. And I know there's a local Malaysian success story here.
Yeah.
Why should you care about this local capability?
The impact is profound. It accelerates healing, it minimizes infection risk, and it significantly reduces long-term scarring. It just drastically improves the quality of life for patients. But the local capability story, that's about healthcare independence and cost.
Ah, okay.
The National Tissue Bank, which was established at USM back in 1991, has been producing grafts since '93. It's benefited well over 1,500 patients.
Right.
By being able to produce these high-quality grafts locally, using local donors, we dramatically reduce the costs. We avoid the massive expense of importing them.
So it democratizes the care.
It really does.
Is there a specific example that really brings this home for us?
Yes, there's a very powerful one from 1993 early on. A young boy had suffered severe scalding from boiling water. Thanks to the immediate work of the surgeons and the supply of what's called an amnion membrane, a type of dressing provided by the National Tissue Bank, his wounds healed beautifully. The scars today are minimal. It showed right away that local, rapid access to these materials could change lives. And this same infrastructure also provides things like bone allografts, which are critical for orthopedic and dental surgeries.
That story really highlights how engineering translates directly into human outcomes. Okay, let's transition from surfaces then to deep internal structure. Let's talk about bone. Unlike skin, the skeleton has a pretty limited capacity to regenerate after major trauma. And traditional grafts, they often require a second surgery, right? Creating what's called donor site morbidity. How does tissue engineering step in to become the ultimate repair crew for bone?
It's a perfect segue because we're moving from a surface to engineering a functional three-dimensional replacement. The solution for bone involves using scaffolds that are seeded with mesenchymal stem cells, or MSCs.
Okay, MSCs.
Right. And if donor site morbidity is the problem,
Yeah.
the pain and complication of taking bone from another part of the body,
Mhm.
then using a patient's own MSCs on an engineered template is the solution. The scaffolds are the bedrock. They're made of biocompatible materials, providing both immediate mechanical support and, critically, a structured environment to guide the growth of new natural bone.
That makes sense. You need the frame before you can start building. But what's the real breakthrough here? I mean, for large-scale bone repair, the biggest hurdle isn't just the cells or the scaffold, it's getting a blood supply, right? Vascularization. How does this interplay overcome that?
That is the essential, difficult question when you try to scale these up. The dynamic interplay, it addresses this by ensuring guided organic regrowth, not just a static replacement. The MSCs, they act like an intelligent construction crew. They differentiate into specialized bone-forming cells, called osteoblasts. And the scaffold, it isn't just sitting there. It's an active guiding template, influencing the spatial arrangement of the new bone. And for vascularization, the scaffold can be designed to be porous. This allows capillaries to grow in. And the MSCs themselves release factors that encourage blood vessel formation right alongside the new bone.
So it's a cooperative process.
It's absolutely vital. It ensures structural integrity and seamless integration, minimizing the chance that the new bone fails from a lack of oxygen or nutrients.
It's incredible to think of those specialized cells and materials working together like that. So let's broaden the scope even further. The ultimate goal, artificial organs. The scarcity of donor organs is a global crisis. What specific complex organs is tissue engineering targeting right now to provide viable alternatives to transplantation?
Well, the field is expanding very rapidly. We're seeing
Yeah.
incredible progress with things like a bioartificial kidney designed with functional renal tissue, and also the bioartificial pancreas, which is capable of precise, regulated insulin production.
And these engineered solutions, they could eliminate the risk of rejection.
That's the hope, to eliminate rejection and the need for lifelong immunosuppression.
Okay, so before we drill down into the pancreas, we have to talk about the infrastructure for any organ. The plumbing, the blood vessels, I mean, they're under intense pressure, they face the risk of clotting. How on earth do you engineer a replacement vessel that's both mechanically stable and biologically non-thrombogenic?
The engineering challenge is, it's extreme. The replacement has to perform flawlessly under constant dynamic forces. But we've seen a lot of success using novel methods to engineer what we call autologous, non-thrombogenic, in situ tissue engineered blood vessels, or TEBVs for short.
Autologous meaning from the patient's own cells.
Exactly, which dramatically reduces rejection. Research in goat models, which are a good analog for humans here, has shown really promising results for arteriovenous grafting. These TEBVs, they achieve the necessary vascular phenotype and, importantly, showed spontaneous endothelialization.
Meaning they grow their own smooth inner lining.
Correct. A lining that prevents clots from forming.
And what about the strength? You mentioned withstanding pressure.
Right, you need guaranteed structural integrity. These TEBVs successfully contained contractile smooth muscle cells, which provide elasticity, and they achieved a very high burst pressure. The documented capacity is somewhere less than 2,000 millam/Hg.
2,000. When human blood pressure rarely gets above 200.
Precisely. It's a huge safety margin. It indicates mechanical stability far beyond what's needed. And this allows them to stay open, maintain patency at rates similar to traditional synthetic grafts, but without the synthetic drawbacks.
That mechanical tolerance plus the biological integration, that is the sweet spot. So what does this all mean for someone, say, struggling with type 1 diabetes? Let's drill down into that bioartificial pancreas, specifically the islet sheet technology.
The bioartificial pancreas, or a BAP, using this technology is a monumental effort to restore the endocrine function of the pancreas. The whole design has to achieve one central goal: let insulin out, but keep the immune system out.
A very selective bouncer at the door.
A very selective bouncer, yes. The system has three distinct engineered components. First, you have the functional element, the encapsulated islet cells. These are the clusters that sense glucose and release insulin, amylin, and glucagon. The encapsulation is the key engineering trick. It's a shield against the patient's own autoimmune system.
Okay, that's part one.
Second, surrounding those cells is a semi-permeable material.
Yeah.
This barrier is highly selective. It's designed to let small molecules like glucose and nutrients in and the hormones out, while physically blocking the large immune cells that would destroy the implant.
And the third part?
Third is the biocompatible outer layer. This layer, often reinforced with synthetic fibers for strength, is critical for survival. It makes sure the implant integrates without triggering a severe inflammatory or fibrotic response from the body's tissues.
It sounds like this incredibly complex piece of architecture, all designed to trick the immune system while performing a really sensitive metabolic job. What is the ultimate long-term promise this holds for a patient once it's implanted?
The promise is restorative autonomy. Once it's surgically implanted, the islet sheet is designed to sense glucose levels and behave exactly as a healthy pancreas should, providing precise, regulated hormone release. The expectation, based on all the ongoing research, is that these sheets will remain viable for years.
For years.
Offering patients the potential to live completely free from the daily burdens of insulin injections and constant glucose monitoring.
This has been an incredibly insightful deep dive. We've moved across the entire human body, from bioengineered skin that revolutionizes wound care to scaffolds guiding new bone growth, and finally, this extraordinary promise of engineered organs like the bio-artificial pancreas. Our utmost gratitude goes to technologist Dr. Rama Yusvana, senior lecturer from the Faculty of Engineering Technology at Tun Hussein on University Malaysia, UTHM. Thank you so much for sharing your invaluable insights and just breaking all of this down for us today.
My pleasure. Thank you for the insightful conversation.
And to leave you with a final, provocative thought. Given how successful we're becoming at engineering the structures and functions of these complex tissues, what's the most critical challenge that remains? Specifically, in integrating these new organs with the body's native infrastructure, connecting them seamlessly to the existing neurological and vascular networks, that's something for you to mull over or explore on your own. We'll see you on the next deep dive.